Good evening to all the NEET aspirants 2026. This is your Prasanna Ma'am from NEET World, and here we are to discuss the 100 concepts, top most important concepts for NEET 2026. So here we are on the last segment of this chapter, cell cycle and cell division, and the topic is meiosis. Let's get started without much delay. So first, in meiosis, before entering the cycle, let me give you the framework of meiosis beta. As we have discussed in our first class of cell cycle, meiosis is being having two phases. Okay, we call it as meiosis 1 and meiosis 2, right ma? So we have meiosis 1 and meiosis 2. Meiosis 1 also will be having what? Karyokinesis and cytokinesis. Meiosis 1 will be having what? Karyokinesis and cytokinesis. Karyokinesis, as we all know, it is divided into 4 phases. What are the beta? As it is meiosis 1, we have Prophase 1, Metaphase 1, Anaphase 1 and Telophase 1. The most important segment here is 5 substages of Prophase 1. In every alternate year of NEET, there is a definite question on this 5 substages of Prophase 1. Mostly if you look into the previous questions, from meiosis if there is a question, mostly it is from Prophase 1 of these 5 substages only. So what are these 5 major substages of Prophase 1 ma? The 5 major substages of this will be leptotene, zygotene, pachytene, diplotene and diakinesis. These are the 5 substages of Prophase 1. And then as we have discussed cytokinesis 1, it will be same as that we have discussed in the cytokinesis of mitosis in both plant cell and animal cell. After cytokinesis 1, in each cell you can look into the univalent chromosomes. Univalent chromosomes with nuclear membrane, nucleolus, everything. I have drawn like this, but univalent chromosomes with chromatin material, nucleus, nucleolus, completely it will be there as such. Okay. This is for cytokinesis 1. After cytokinesis 1, we have interkinesis. Interkinesis, or this phase we will also call it as intermeiotic phase. This phase is interkinesis or intermeiotic phase. Intermeiotic phase. In intermeiotic phase, there will not be any DNA replication, but proteins and RNA, whichever is required for meiosis 2, will be synthesized. And third most important point is, if it is an animal cell, centriole duplication will also happen in interkinesis. Let us discuss in more detail when we finish this karyokinesis 1 and cytokinesis 1. Here in meiosis 2, again we have 2. What are they? Karyokinesis 2 and cytokinesis 2. Meiosis 2 is same as that of mitosis only. Meiosis 2 is same as that of mitosis beta. Okay, and karyokinesis, as we all know, it has 4 substages as usual. Those we call it as what? Prophase 2, Metaphase 2, Anaphase 2 and Telophase 2. That's it. Okay, and here if you see meiosis, this is also called as reductional division. Meiosis is also called as reductional division. Why is it also called as reductional division? Yes, in daughter cells, if you see from parental cells to daughter cells, in daughter cells chromosome number will be half. Chromosome number gets half in the daughter cells. That is why meiosis is said to be the reductional division. And this is the complete framework of the meiosis. If this flow chart is in your mind, then you can remember all the stages in the sequence. Now, let us look into the meiosis 1, Prophase 1. Prophase 1. As we have discussed, Prophase 1 will have how many substages? 5 substages. Of that first substage is what? Leptotene. Leptotene is the first substage. What are the key features of leptotene? What do you observe in leptotene actually? In leptotene, you can see these thread-like chromatid structures, and the movement of asters you can see, centriole movement, asters movement you can see. In centrioles also, see, I will tell you in detail. If this is centrosome, can you see the centrosome? These two are the perpendicularly arranged structures, centrioles like this. They start moving to the poles in the leptotene. Second most important stage is what? Chromatids. Chromatids will be formed, but these chromatids will be attached to the nuclear membrane. And how the chromatids will look like? A bouquet, complete bouquet it will look like. So, you will call this stage as bouquet stage also. Okay, that is due to the structural visualization of chromatids. And here in the leptotene, you can observe the chromatids clearly under the light microscope. Fine, ma. This is all about leptotene. In zygotene, what happens is, those chromatids which have same alleles over them at same location, we call it as homologous chromosomes. So, here you can see synapsis concepts or synaptonemal complex formation or homologous chromosomes coming together. Before that, you should be clear on the notion of homologous chromosome. You should be clear about what is homologous chromosome. Homologous chromosomes are nothing but, see, chromatids are formed in the leptotene, right? In those chromatids, in the complete genetic material, what is present? Kunal, you say, what is present in the genetic material or these chromosomes? DNA. Yes, DNA gets condensed to what? These chromatid structures, right? DNA gets condensed to this chromatid structures. In this DNA, what is present? In this DNA, what is present, beta? Don't mix up everything. In this DNA, what is present? Genes are present. What do you call a gene as? Inheritance, okay? It is the unit of inheritance. A gene is a combination of two alleles. We have learnt in principles of inheritance or genetics. It has two alleles. So, these two alleles at same location forms a gene, okay? At same loci, two alleles are present, will form what? One gene. As such, these are now called as what, beta? Homologous chromosomes. Homologous chromosomes are nothing but the alleles located at the same loci. Tell me any character, hair color or eye colors or height of a human being's skin color, any character. Tell me one character among that, Kunal. Hair color. Few hair color texture will be soft, sometimes it is black in color, sometimes it is brown. Let us assume black is dominant, brown is recessive. Hair character, that is one character, capital B and small b. These two alleles with respect to one gene will code for one phenotype, right? So, they are at same loci, these two chromatids will come together and form what? Homologous chromosomes. As such, you will be having chromosomes like this. These two are homologous chromosomes with capital B, small b. Tell me some more features like skin color. Skin color with maximum melanin pigment is dominant, right? So, that let us assume capital M and small m. Same location, right, beta? So, these two are what? Homologous chromosomes. So, formation of homologous chromosomes. Homologous chromosomes. Okay. Homologous chromosomes. Here you can see bivalent. Bivalent means two chromosomes. So, in one synaptonemal complex, association of homologous chromosomes is called as synaptonemal complex. In this synaptonemal complex, two chromosomes are present. Okay. So, you call this two chromosomes as what? One bivalent. In this one bivalent, how many chromatids are present, beta? How many chromatids are present? 4 chromatids are present. 1, 2, 3 and 4. 4 chromatids means it is tetrad. One bivalent is equal to one tetrad. One bivalent is equal to one tetrad. and this association of synaptonemal complex is simply termed as what? Synapsis. So, any time you come across the terms beta, synaptonemal complex, bivalent, homologous chromosomes, you have to choose which substage? Zygote in substage. And here in this homologous chromosomes, you should be clear with sister chromatids and non-sister chromatids. These two chromatids of the same chromosome are said to be what? Sister chromatids. These two chromatids of different chromosomes are said to be what? Non-sister chromatids. These are non-sister chromatids and these are sister chromatids. Clear, right, ma? So, you are clear with leptotin stage and zygotin stage, right? Third most important stage of prophase I, okay, is what? Pachytene stage. So, in Pachytene stage, in between that homologous chromosomes of non-sister chromatids, okay, recombination happens. Okay, what happens, ma? Recombination in between two homologous chromosomes of non-sister chromatids, which fine, recombination occurs between two homologous chromosomes of non-sister chromatids. Takes place in which phase? Pachytene phase. And this recombination is also called as what, Nana? Crossing over. Okay, this is also called as crossing over. Fine, this is also called as crossing over. Recombination or crossing over is an enzyme mediated process. Do remember, this is an enzyme mediated process. What is that enzyme that is mediating? It is an enzyme mediated process, and the enzyme that is mediating is what? Recombinase. Recombinase is the enzyme that is mediating the recombination between two non-sister chromatids of homologous chromosomes. Till here it is clear, no beta? Next, fourth most important substage, that is diplotin. Once after crossing over is finished, what happens? Yes, it forms an chiasmata, X-shaped structures. Okay, can you see here the X-shaped structures clearly? See, this is what? Crossing over point, crossing over point. Apart from that crossing over point, rest chromosome will come apart from each other, which gives rise to what? X-shaped structures. These X-shaped structures are called as what? Chiasmata. So, where do you see chiasmata, ma? Chiasmata or X-shaped structures you will observe in which phase? Diplotin phase. This is fine, no? Next most important point, that is dissociation of synaptonemal complex. The synaptonemal complex which has formed is dissociated in diplotin by formation of what? Chiasmata. Synaptonemal complex is like this. Now that has become like this, X-shaped chromosomes. So, here one more point is what happening, like dissolution or synaptonemal complex. Okay. Dissolution of synaptonemal complex. Clear, ma? Dissolution of synaptonemal complex, right? Clear with till here, right, na beta? So, in diplotin what is happening? Chiasmata and dissolution of synaptonemal complex. Here one point I have to deliver, that oocytes of most vertebrates and invertebrates will stuck in this stage of meiosis for years together. And like in menstruation cycle, every cycle one egg will be deposited. That is through what? Many eggs will be formed in the embryonic stage itself of a female. That cell division is being stuck at which phase? Diplotin. Prophase I of diplotin. Okay. So, here what happens, ma? Oocytes of vertebrates and invertebrates will stop their cell division at diplotin substage of prophase I. Till here it is fine, na beta? Pachytene and diplotin. Now, the last substage of prophase I. the last substage of prophase I is diakinesis. In diakinesis, dissolution of chiasmata will happen. Okay. In diplotin, what's happening? Dissolution of synaptonemal complex. But in diakinesis, dissolution of chiasmata happens. Okay. So here in diakinesis, dissolution of chiasmata takes place. And here in diakinesis, nuclear membrane, nucleolus, endoplasmic reticulum, Golgi bodies will disappear. Okay. Complete disappearance of nuclear membrane, nucleolus. Okay, nuclear membrane, nucleolus and endoplasmic reticulum, Golgi will happen in the diakinesis. This is all the 5 substages of prophase I. Let us recall quickly. In the leptotin stage, what's happening? Bouquet stage is formed. Okay, where chromatin thread-like structures will be formed, right? Condensation. In zygotin, synapsis, synaptonemal complex, formation of bivalent, tetrad condition is observed. We can clearly see sister chromatids and non-sister chromatids. Third phase, that is pachytene, crossing over will take place. Okay. In crossing over, that is an enzyme mediated process by the enzyme recombinase. Crossing over happens between non-sister chromatids of homologous chromosomes. Third is also fine, beta. Fourth, that is diplotin. In diplotin, 2 major points or events that you have to remember. What are they? Dissolution of synaptonemal complex, and oocytes of vertebrates and invertebrates will be stopping their cell division. They will cease the cell division in this substage of prophase I. And X-shaped structures are observed. Formation of chiasmata, dissolution of synaptonemal complex. And next is what? Last substage, that is diakinesis, wherein dissolution of chiasmata is seen, wherein nuclear membrane, nucleolus, endoplasmic reticulum and Golgi bodies will disappear. Now, in metaphase I, here you can see, not same, here you can see some differences in meiosis I. You can see 2 centrosomes at the opposite side. You can see centrioles also radiating out the spindle fibers. They will connect to what? Kinetochore, which are disc shaped structures over the centromeres. Here spindle fibers will be connected. One major difference you will see is here you can find bimetaphasic plate. As you have homologous chromosomes, 2 sets of chromosomes, right? So you can observe what? Bimetaphasic plate. And this all chromosomes coming at the center, you will call it as what, Nana? Congregation. Congregation. Well, all chromosomes will come at the equatorial plate, and here in metaphase I you can see bimetaphasic plate. Bimetaphasic plate. Yes. Okay. So this is all about metaphase I. Next is anaphase I. In anaphase I of meiosis and anaphase of mitosis and anaphase II of meiosis, there is a difference. What is that? Can you say, Kunal?
Whole chromosomes are pulled towards the pole.
Of course, that is true, but that we can say in a single sentence, like there is no centromere splitting over here, beta. Okay, there is no centromere splitting. Centromere splitting is observed in anaphase of mitosis and anaphase II of meiosis, but in anaphase I there is no centromere splitting, wherein the chromosomes which are aligned with above the equatorial plane, about first equatorial plate, those will move towards the upper pole, and the chromatids or chromosomes which are at the lower equatorial plate, they will move towards the lower pole. There is no centromere splitting in the anaphase I. And next, telophase I, same case, but here you can see dyad condition. Dyad means what, Nana? 2 chromosomes you can see in a single nucleus or in a single cell, where tangling of the chromosomes into the chromatin will happen. Okay, chromosomes will get tangled to chromatids. Chromosomes will get tangled to chromatin. Chromatin. and you can see reappearance of what? Nuclear membrane. Very good. First you have to say nuclear membrane and nucleolus beta. Then you can also see the Golgi and ER. This is what the changes that you can observe in telophase 1. Clear, right? Now, so before entering into the meiosis 2, meiosis 1 is clear, right? And this phase in between meiosis 1 and meiosis 2 is what? Interkinesis. Interkinesis, or you can also call it as intermeiotic phase. Intermeiotic phase. In intermeiotic phase, there is no DNA replication. There is no DNA replication. Only there is no DNA replication. But there is centriole duplication in animal cells. Okay. Centriole duplication in animal cells. Fine. And there is one more event, that proteins and RNA, whichever is required for meiosis 2, are being synthesized in this intermeiotic phase. Okay. Proteins and RNA required for the synthesis of meiosis 2 are being synthesized in the intermeiotic phase. That is it. These are the 3 clear points that you have to remember better. In meiosis 2, one small note, that meiosis 2 is same as that of mitosis. That is it. Meiosis 2 is same as that of mitosis. So, in here if you see, we all know that here you have prophase 2, metaphase 2, anaphase 2, telophase 2. As you know mitosis very clearly, you tell me in prophase 2 what happens?
Prophase 2, condensation of chromatin into chromosomes.
Yes, you can also call it as untangling of chromatin into chromosomes or condensation of chromosomes. Fine. Second point?
Centrioles go towards the poles.
Yes, first is condensation. Second point is what? Centrioles. Yes, very good. Formation of aster fibers, and then spindle fiber apparatus, and then nuclear membrane, nucleolus, everything gets endoplasmic reticulum, and Golgi bodies. Everything will get disappeared. In metaphase 2, if you see,
Spindle fibers get attached to.
In metaphase 2, uniequatorial plate, single equatorial plate is formed as a result of congregation. Equatorial plate is formed as a result of congregation. Clear? So, here you can see that equatorial plate. Okay, and spindle fibers will be extended from the asters to attach to the kinetochores. In anaphase 2, centromere splitting will happen, same as that of mitosis. Okay, centromere splitting happens. Centromere splitting. Telophase 2. Formation of? Yeah, that happens in animal cells. That is during cytokinesis, but not telophase 2. In telophase 2, again they get tangled. Chromosomes get tangled into what? Chromatin, nuclear membrane, nucleolus, everything gets reappear. Okay, this is about what? Meiosis 2. Clear, na? So, here splitting of chromosomes will happen in which phases? Anaphase of mitosis, anaphase 2 of meiosis. Do remember, and then everything is same as that of mitosis. Okay. Now, let us exercise few questions as we are clear about cytokinesis of plant cell and animal cell. We have discussed in the previous class only. So, look into the major differences between mitosis and meiosis first. What are the major differences between mitosis and meiosis, beta?
Somatic cells.
Somatic, not stomatic. This occurs in somatic cells, but meiosis occurs in germ cells. And here nucleus divides once. Here how many karyokinesis happens? 2 karyokinesis. And one major difference, 2 daughter cells. Here how many daughter cells will be formed? 4. Daughter cells here, ploidy, yeah, ploidy is same as that of parent cell. Ploidy is same as that of parent cell. If parent cell is diploid, it will also be diploid. If parent cell is haploid, this will also be haploid. Whereas in meiosis, daughter cells ploidy will be haploid. This occurs more frequently because complete system of living body runs by mitosis only. Okay, but here this occurs less frequently, only at the time of fertilization. Daughter cells will form somatic organs through this. They form gametes during gametogenesis. Next, there only one set of karyokinesis phases like prophase, metaphase, anaphase, telophase, only one set will be there. But here you can see 2 of each phases and 5 substages of prophase 1 will be there beta. Next, number of chromosomes are not changed, but here they are reduced to 8. No crossing over, but here crossing over occurs, and here equational division. Mitosis is also called as what? Equational division, meiosis, reduction. Yes, yes. Now let us exercise few questions so that you will also get confidence after listening the class. How many chromosomes will the cell have at G1 phase, S phase, and what phase is asked beta? M phase. Respectively, if it has 14 chromosomes at interphase. See, interphase is a combination, or it has in interphase, what does it have? G1, S and G2, and M phase is mitotic phase. He said that how many chromosomes will be having, if at all 14 chromosomes are there in interphase, he said. That is, in G1 phase it has 14, in S phase it will be 14, in G2 also it will be 14. Listen, in G1, S and G2 it will be 14 chromosomes, because in S phase there is no chromosome duplication. There is only DNA duplication. Okay. In M phase you can see the chromosome. See, what do you see? 14, 14, and in M phase also chromosome will be 14 only, because in nowhere in the question he has mentioned meiosis. After M phase, chromosome number will remain same. If at all he is asking after meiosis, then your answer will be 14 in the G1, 14 in the S phase, after S phase, and in M phase it will be 7. If he is asking you meiosis, he just said M phase. In M phase, the option that fits is 14, 14 and 7. Clear, right? Okay. So, that is why, though you know concept, though you have complete knowledge about this concept, sometimes you will make it wrong because you are in hurry of answering the question. Look into the question properly beta, there is no hurry. Okay. This question you see ma. This question you look into. How many meiotic and mitotic divisions need to occur for the development of mature female gametophyte from the megaspore mother cell in an angiospermic plant? This question is in connection with sexual reproduction of flowering plants. This question, NEET 2025 question. This question is in connection with NEET 2025 and the chapter that is sexual reproduction of flowering plants, where connecting the cell division he has asked megasporogenesis. Okay, those two concepts are connected. Look into that properly. See, if I have to draw an ovule and explain you clearly, okay, inside this ovule completely cells are present, like how in mother's womb, placenta is present. This inside this ovule complete cells are present. What are they, Nana? Nucellus. Nucellus cells are diploid. One of the nucellus cells in this micropylar region gets differentiated into megaspore mother cell. He has asked from megaspore mother cell, no, so we have to start counting the division from megaspore mother cell only. This undergoes one reductional division forming 4 megaspores. These are haploid megaspores will be formed. Of this 3, of this 4, 3 will be degenerated and 1 megaspore which is towards the chelazal end, this is towards the chelazal end, this will be functional. okay, and this one cell will undergo 3 sequential mitotic divisions beta. 3 sequential mitotic divisions means 1 and 2, fine, this is one division. Second division, it will form 4 cells over here, 4 nuclei over here and 4 here. After third, you can see 4 at each pole, okay. From here, one nucleus from the chelazal end, one from the micropylar end will come at the center. As such, the embryo sac is formed, okay. 3 antipodals at the chelazal end, egg apparatus with 2 synergids and an egg cell at the micropylar end, and at the center you will be having polar nuclei. Very good, okay. Polar nuclei also called as secondary nucleus. Fine. This embryo sac is what? Female gametophyte of an angiosperm. Fine. This female gametophyte of an angiosperm is formed from what? Megaspore mother cell. After 1 meiosis and 3 mitotic divisions. This is how you have to learn the concept. Okay. Connecting the dots, connecting the concepts of different chapters wherever required. That is how you have to learn. Okay. So, here what will be the right answer, ma? 1 meiosis and 3 mitotic divisions. Clear, right? So, with this we will end today's session. Fine, we have done enough practice and enough explanation for this small topic. From tomorrow we will be starting biomolecules, okay.